Hello everyone, my name is Josh and today I'll be talking to you about my PhD project, which is using environmental DNA to detect flower visiting insects for avocado trees in the Southwest. So this project is with the South West Catchments Council as well as Horticultural Innovations, and it's being done at two universities, Curtin University and Edith Cowan. So what is environmental DNA? Well, effectively, it's the trace amounts of DNA left by organisms on a substrate of interest. In this case, we're interested in insects and the DNA that they leave on flowers. So if we look at the example of our little bee here, when it visits a flower, it interacts with the pollen, it interacts with the nectar, and it touches all these floral surfaces. And in doing so, it leaves traces of DNA on that flower, which I've represented here as the blue dots. We can take this flower as a sample, or in the case of avocados, which are mass flowering species, we can take an entire inflorescence. And that inflorescence can form our sample. We can then extract the DNA from that sample and amplify up just the insect DNA using insect primers. The sequences that we then generate are then compared to online reference databases, which contain sequences from organisms that other people have studied, sequenced, and uploaded to those online databases. If the sequences that we generate match the sequences that someone else has uploaded onto the online database, then we have the potential to get species identification. In the case of this example, you can see that the species identification would tell you that it was a honeybee that was visiting that flower. Now, why would we want to use environmental DNA for avocados? Well, the first question that comes up within the avocado industry is, who are the significant pollinators for avocado trees? After all, avocado trees are native to South America, where they're pollinated by stingless wasps. But we don't have those stingless wasps here. And as we know from the introduction of cane toads, doing species introductions across different countries is not such a crash hot idea. So instead, we're left with what we've got, which is wild pollinators, such as native bees, native wasps, and flies, but also managed pollinators, i.e. the European honeybee. But who's doing the most significant significant load of doing pollination services? Well, that remains a huge question for the industry. Now, how these pollinators, both wild and managed, vary in space and time is also a big question. So by time, I mean across the avocado flowering season. Do you find, for example, the pollination services are low at the beginning of the avocado flowering season, high during peak, and then tapering off as the avocado flowering diminishes, or is it consistent throughout? On top of this, how do pollinators vary within the space of an avocado orchard? If you're at the edge of an orchard, do you have different pollination services and therefore different yield than at the center of the orchard, where the edge effects from being adjacent to pasture, bushland, etc., maybe are a lot weaker? On top of this, there's a question of, what does co-flowering resources do for avocado pollination? If you have bushland that's adjacent to an avocado orchard and it's flowering at the same time as avocados, do you find, for instance, that the avocado orchard is getting reduced pollination services because your insects are way more interested in getting into the native vegetation surrounding the orchard? Or do you find that having more flowering resources in the landscape actually increases the diversity of insects, meaning that you might then get more flower visits for your avocado trees and potentially greater yield? So ultimately, the question that we have is, can environmental DNA be used to classify these flower visiting insects and then determine who the most important pollinators are? So the key objectives of this study are to determine, firstly, the flower visitors using environmental DNA. Second, to work out of these flower visitors, who are the most important pollinators. And then lastly, to see how these pollinators vary through space and also through time. So stage one of this project was asking that first fundamental question. Can we use environmental DNA to determine the flower visiting insects for avocado trees in the Southwest? We did this work in two orchards, Marronbrook Farm in Middlesex and Bendotti Avocados in Pemberton. Now, the idea with this first study was, if we compare environmental DNA with two tried and tested methods and it agrees with them, then that gives us the confidence that this technology can work. So what did we compare? Well, environmental DNA is our one at the bottom. In the middle, we have pan traps, which are tried and tested to capture the general diversity of avocado flower, to capture the general diversity of insects in an orchard. And then the third one is cameras to monitor flower visits to avocado trees. And like I mentioned previously, we were interested in looking at the congruence between these three methods. Do you find, for example, that cameras detected these two flower visiting insect species and that eDNA found exactly the same ones? Or maybe cameras captured two, but eDNA only captured one. suggesting that this might not be the most effective technology to measure flower visiting insects. So what did we find? Well, firstly, I just wanted to show you a quick video that I captured in one of the orchards down in Pemberton. And I think for me, coming from a natural sciences background, the most pleasant surprise was just how much diversity is intrinsic within these avocado orchards. They are alive with activity. So many different types of flies, insects, bees, moths. It's phenomenal. And so they really are a huge refuge for native insects. And so if we have the potential to harness the pollination services that these native insects offer, there's huge benefits for the industry. So let's compare the methods. So what did cameras find? So cameras, they're really good at detecting the big things that visit avocado flowers. These are the big bush flies, ladybugs, wasps, and moth species. But the diversity is much lower when you compare it to pan traps. They capture everything from grasshoppers to wasps to gnats and to beetles as well. But the caveat with pan traps, and a really interesting point, is that they capture everything. That's not just flower visiting insects for avocado trees. And so this diversity is somewhat inflated that you see in the pan traps. Things like grasshoppers, we know that they're not avocado flower visitors, but we also know that they're definitely not pollinators. So this comes with a lot of caveats. And lastly, what about eDNA? So eDNA is really good at picking up the small things that spend a lot of time on the inflorescence. These are the gnats, the small flies, as well as thrips. But most importantly, eDNA agreed with cameras and pan traps that the most common flower visitors and most common insects within the orchard were honeybees and hoverflies. And for anyone in WA that remembers the 2020 season, hoverflies were prolific. You could absolutely hear the drone of them in the orchard. There were millions of them. So the fact that eDNA agrees with cameras, which agrees with pan traps, that these are the most common flower visitors, gives us the confidence to say that environmental DNA can be used as a technology to measure flower visiting insects. And that gives us really exciting potential for the future when we're going to do it in future studies in order to use this to measure flower visiting insects and compare different orchards to see how the community varies in space and time. So stage 2 of the project, this is a little bit different. Now we're interested in how does insect diversity and the diversity of the pollen that they carry vary across the avocado flowering season, depending on if orchards are adjacent to pasture or bushland, which are the two most common adjacent land uses in the Southwest. So here we're using a similar technique to the one I showed you previously, but with a little riff on it. So we're interested still in the fact that these insects are visiting avocado flowers, but instead of targeting the insect, we're actually interested in targeting the diversity of the pollen that the insects are carrying. So once we wash the insects and we get the pollen off, we can take that as our sample. And once that sample is transported back to Curtin, we can take it through the same workflow that I showed you previously. The difference here, though, is that instead of using insect primers, we're using plant primers. The plant primers can amplify up our DNA of interest, which again, we sequence and then we compare to the online databases, which can then get a species identification for the different types of pollen being carried by these insects. What's interesting about this is that we can determine who the co-flowering species are that are visited commonly by avocado flower visiting insects that are important potentially for pollination. The idea being that if you can increase the resources for avocado pollinating insects, then you might be able to increase their numbers, diversity, and hopefully visits to avocado flowers and potentially yield. So the way we measured the insect diversity within these orchards was through pan traps. Pan traps were set up in the orchard as well as the adjacent land uses for these orchards. And we targeted 6 orchards in total. 3 were adjacent to pasture and 3 were adjacent to native bushland. And we were interested in measuring this at 4 different time points. So the first time point that we targeted was before avocado flowering had begun. That's our baseline measure. We then compared this to beginning to see as flowers are starting to emerge, what's happening with the community. Then peak, which is when it's busy, lots of activity, heaps is happening. And then finally after avocado flowering to see if it returns to the baseline of before or if it's slightly different. So the overall objective with this study is that we're really trying to determine the landscape factors and the co-flowering resources that most strongly influence the insect diversity and also the diversity of the pollen that they carry. Like I mentioned previously, the reason this is interesting is because we want to work out what resources benefit avocado flowering insects, aside from avocado trees themselves, to see if we can help plant these within an orchard or plant native species around an orchard potentially, to help prime that community so that when avocado flowering is at peak, the insects are there and they're ready to go without necessarily having to just pay extra money to hire more hives, which can be a huge expense. And so that's really what we're trying to target with this study. Now, I wish I could give you results, but unfortunately, it's only one PhD student on this project at the moment, and that's me. And so at the moment, I'm actually at Curtin University processing these samples and taking them through that workflow that I mentioned previously, extracting the DNA, amplifying it up, sequencing it, comparing it to the databases, and getting those species identification. It's a slow process at the moment, a bit of a slog, but I'm making good traction, and I look forward to sharing those results with you shortly. But in the meantime, I just want to thank you all so much for listening. I hope it wasn't too confusing, and if anyone's got any questions, please shoot away. The last thing I'll say is a big thank you to my volunteers in the 2020 field season. That was my mum and my 92-year-old granddad. I honestly couldn't have done it without them. They were lifesavers. So thank you to them, and thank you again to you for listening.
Thanks, Josh. Back over to you, Wendy. I think you might ask if there's any questions out there. I don't think anybody has got their hands up at the moment, but somebody might want to just unmute and ask a question.
I think I got a piece.
Oh, hang on.
I think somebody's going to unmute.
Yeah, that's me.
Oh no, here we go.
It's just one question.
Sure.
It's more of an observation during your work than
Yeah.
actually what you're doing, because obviously you're waiting on the results.
Yes.
But I was just wondering, did you notice much of a difference with cloudy days and sunny days and honeybee activity as compared to some of the other stuff? Just sort of an off-the-cuff observation, maybe.
Oh, absolutely. It's quite extraordinary. On the sunny days, honeybee activity is a lot stronger, but also just the sunny, warm days. It just has a huge influence. The 2020 field season in particular in the Southwest was a nightmare. It was really wet, really cloudy. And so when we could get those sunny days, it was such a stark contrast in terms of activity for not only honeybees, but also the native insects as well. Yeah, they're so much more active. And I think that has something to do with the fact that the flowers, how visible they are to honeybees is a lot stronger when the sun's out compared to when it's cloudy. And so I think that they have they're a lot more in tune with the whole system as well. When it's really bright, really sunny, it's all happening. And I think, yeah, I couldn't believe how stark it was. But I don't have the data to back that up at the moment, but just from my observations, completely, if it's warm, sunny day, honeybees are happy. But yeah, but if it's cool or it's cloudy or it's wet and it's windy, you just see that activity definitely go down quite a lot.
Thanks heaps.
No worries.
Thanks. I think Carly has got a question as well.
Yeah, just talk. Can you hear us?
Yes.
Yeah. Oh, cool. Go, Genesis.
Oh yeah, I've got a question. I know the results aren't in yet, but what's your intuitive feeling? I know you're not, as a scientist, you're probably not supposed to have preconceived ideas about.
Oh, go in so many biases. Keep going.
Are you expecting sort of that the properties, the avocado orchards next door to native bush are likely to have more pollinators and thus better pollination all year round, or during the flowering season?
Great question. So what's really interesting is when I was setting up the pan traps and then collecting them, you get a count of how many insects you've got in these pan traps, and so you're starting to get an idea of the diversity. And what's extraordinary is in the bush, there is just so much happening. There is so many native bees. There's a lot of insects. There's so much. The difficulty, I think, is that are avocado trees interesting enough to pull the insects away from the native bush into the orchard? And so what's interesting is you'll see, for example, I think I saw in one kind of sampling period for bush, there was about 30 native bees just in that sample. But when I went to the orchard, there was one in that whole area. And so it's the transfer of insects from that adjacent land use that I'm not convinced is actually happening at the moment. And I think that's probably because obviously the native bees have evolved alongside these native plants, whereas avocado trees are pretty new to the show, and the amount of pollen and the amount of nectar that they're generating would be so much less than the native plants. And so, yeah, that's why I really don't know at the moment until I process this what's going to happen, because I think at the moment, is native going to result in high pollination? I hope so, because I would love to hopefully put a monetary value to native bush and say, let's not clear it, because, you know, we don't need to just keep expanding avocado orchards. We actually need to keep this in the landscape. But I'm not convinced at the moment that that there's the pull to get avocado pollinators out of the bush into the orchard. So I hope that answers your question.
Yeah, and there is European bees in the native bush as well, isn't there?
Yes, there is, absolutely. But compared to the native bees, they're quite reduced in number. But I was actually surprised that there were more European honeybees in the bush. I expected to see like quite large numbers, because when I've talked to growers down in Pemberton, the general consensus is that if there's something flowering at the same time that's in the native bush as avocados, then there's just a pull away from the avocado orchard into that native bush. And so I was expecting to see, you know, these traps full of European honeybees. They weren't, though, which was really interesting. So I'm not sure if the consensus is like, you know, like we obviously only sampled in a small subsection, so that's a possibility. But I wonder if that's actually true, that these native bees are being pulled out. And hopefully, as we, you know, interpret these results further and I generate them, we'll be able to kind of unearth that a little bit more to work out, is it actually competition or is it kind of a neutral effect?